Okay, good morning everyone. Nice to see you again. We're looking at this all-important topic of pharmacogenomics in these lectures, and as I think I said previously, I can't claim to be an expert at all in this field, but I thought it was important that we did have some lectures on this topic because it's a very important part of modern pharmacology. So what we're doing is looking at this idea of pharmacogenes, and the pharmacogene is really something that controls the response of the body to a drug. And of course in our last lecture we looked at perhaps the best study of all pharmacogenes, cytochrome P452D6, which is probably more subject to genetic variability than any other gene known to pharmacology. But probably a close second would be SLCO1B1. So this is the topic of today's lecture. And remember, we've been introducing this idea of a VIP, a very important pharmacogene. And I just checked the update this morning. So the PHAR3320, the Pharmacogenomics Knowledge Base, I think based in Stanford University in the US, they have updated their list of VIPs, and I couldn't actually identify the one that's the extra one, but there's one more TL1 VIP than when I last put this lecture together. But we've been looking at the ones that are particularly involved in pharmacokinetics of drugs, and then we'll probably also need a lecture on looking at some of the pharmacodynamic modifying drugs. But the reason I'm focusing on pharmacokinetics, or genes that shape the pharmacokinetics of drugs, is that they tend to affect a range of drugs and not just a small subset that act on a particular receptor. So that's why there tends to be a bit more interest in pharmacokinetic-based genetic variability. Okay, so we've seen that most of the very important pharmacogenes identified to date are involved in metabolism of drugs, and a lot of these are pretty familiar to you, cytochrome P450 enzymes. But then there's the odd phase 2 conjugative enzyme as well, involved in glucuronidation, such as UGT101 and NAT2. But today we want to talk about a transporter, and how that inherited genetic variability in your ability to transport drugs from one compartment to another or via fluid to another can influence the clinical response to a drug. So we're going to focus on one of these transporters, in particular SLCO1B1. Okay, so remember why we've been focusing on genes that control the pharmacokinetic fate of drugs, because they have a lot to do with the concentration of drugs achieved at the target site, at the target receptor. How long that drug sticks around at that site of action has a lot to do with pharmacokinetic variables. Okay, so that's why we're interested in the role of transporters that assist on and run over the top of passive diffusion to bring drugs into their target tissue or excretory organs, whatever tissue we're looking at really. But then there's also efflux transporters removing drugs. And then, of course, within cells, there's often drug metabolizing enzymes carrying out chemical alterations to form metabolites. So the basic point I've been trying to make in these lectures is that inherited polymorphic variants of either influx transporters, efflux transporters, or drug metabolizing enzymes have a big impact on concentrations of drugs at the site of action at their target receptors. So that's why they tend to have a more overriding influence clinically than inherited pharmacodynamic factors such as drug receptors. Okay, so we introduced the importance of transporters way back in first semester, and I'm sure you've probably forgotten all that, but just remember that there are these very important proteins that function like molecular turnstiles to make drugs or help drugs move across cellular membrane barriers. They're important in the gut epithelium, kidney tubules, blood-brain barrier, but the one we're focusing on in this lecture are the hepatocytes And in particular, the one we're interested in is this OAT P1B1 here, and it's a major player in bringing drugs into hepatocytes. So this is important because it has a role to play in ensuring substrate availability to P450 in the liver. Okay, so SL1B1, and it makes it confusing that this is known by multiple names, but it's essentially, its current name is SLCO1B1. It encodes an organic anion transporting polypeptide 1B1, previously known as P2, organic anion transporting peptide 2, and C, also liver-specific transporter 1. So it goes by a number of aliases, but the current recognised name in pharmacogenomics is SLCO1B1. So it's involved in bringing molecules out of the basolateral, across the basolateral membrane into hepatocytes, where they can then be metabolised or become substrates for biliary excretion or excretion across the basolateral membrane back and make their way into the renal excretion pathway. Okay, so this is a pretty important player, pretty important influx transporter. Now, this is a tougher area to teach in a way than some of the P450s, which are quite well characterised proteins these days. Once people figured out how to crystallise cytochrome P450s, we got a lot more information from X-ray crystallography studies of their structure and function. No one's really figured out how to do that with this particular transporter yet. They're very difficult to create X-ray crystals. But also, another factor is that all these transporters that facilitate drug uptake into the liver can often do each other's job. And so sometimes if you have a deficient gene, then it's not that clinically significant because other transporters can do the job and bring drugs into the liver. So you often only see a sort of a narrower clinical impact. It can still be very important if there's a particular drug that there is no redundancy in or aren't other transporters that can bring it into the liver, then you will see a clinical impact. But often it's not always as obvious with other types of drugs, even if they are substrates for that transporter, because they can other pathways can facilitate uptake. Okay, so the high level redundancy with between 1B3 and 1B1, so lacking 1B1 often doesn't have much functional impact for many drugs because 1B3 just does the job. Also, there's no real 1B1 counterpart in lab animals, rats or mice, so this makes it tough to study this protein's function for particular drugs in an animal model. So what you need to do basically is express a human transgene in an animal, create a transgenic model if you want to look at this pathway. Also, there is a very chaotic naming system for allelic variants of SCLO1B1, which made it difficult for people to get their heads around different variant forms and their functional properties. Okay, so you can see so this is a putative computer model. So in underlined putative there, it's based on related proteins where we have succeeded in crystallising them and doing X-ray crystallography studies. People have lined up the sequences that are common across SLCO1B1 and with other homologous sort of genes to try and figure out what the structure might be. So it's basically this multiple membrane spanning domains, a dozen of them, to form this molecular pump structure. Okay, so in perhaps more familiar terms, here we can see this molecule in a more conventional representation. So it's a great big long protein. Both of the termini, the C and the N termini of the protein are located intracellularly. The amino terminus and the carboxy terminus are both located intracellularly. And there's a number of domains, membrane spanning domains, a dozen of them. But then also the linking extracellular and intracellular domains vary quite a bit, but the one that's really distinctive is this whopping big extracellular domain, and some clinically important variants are found in these in this particular large extracellular loop. Okay, but they're also subject to glycosylation, as is commonly the case with extracellular loops. Okay, so a fairly big protein, molecular mass of about 84 kilodaltons, so a fairly good sized protein. Now, why are we talking about this protein and why is it right at the top of the list of very important pharmacogenes? Well, it's really due to the fact that it is important in transporting a whole lot of important pharmaceuticals, and we can see some of them here across a whole lot of very significant drug classes: antibacterials, anti-neoplastic drugs. Ones that really matter, the statins, torvastatin, pravastatin, rosuvastatin, simvastatin, all substrates for this hepatic uptake pathway. But some other important drugs too: methotrexate, very important pharmaceutical, ACE inhibitors, angiotensin II receptor blockers, endothelin receptor antagonists, perhaps not as clinically used, but certainly ACE inhibitors, angiotensin II receptor blockers are mainstays of modern hypertension therapy. So these are significant pharmaceuticals that are and statins as well, for controlling hyperlipidemia. So these are drugs that are used a lot in modern medicine. But if that's not enough, this transport is also important in transporting bilirubin and a number of other endogenous substrates. And this is another level of clinical significance for this particular protein. We'll come back to that in a minute because there are people with inherited deficiencies that show up clinically. Now, just to show one of those drugs I mentioned here, methotrexate. So that's used in cancer chemotherapy, but also in treatment of rheumatoid arthritis. Different sort of dosing schedules, and used in cancer, lower doses and not administered as frequently, but still an important drug. But you can see what happens in animals if you express them with the human gene for 1B1 and administer methotrexate to them. Then the animals with the transgene gene have lower blood concentrations than in the transgenic or than in the wild type animals with the black circles. So the white circles are animals with a functional human SLCO1B1 gene. So basically it's saying that if you put the human gene in the liver of the mouse, it can accumulate methotrexate just like a human. And so we see this in terms of the pharmacokinetic behaviour of methotrexate. The liver concentration goes up significantly, about twofold, in the animals expressing the human transgene compared to wild type animals. So remember I said rodents don't have a 1B1 type transporter, so you can just simply put a human transgene in there and study this phenomenon in a mouse model. Liver plasma ratios are pretty important in pharmacokinetics, and you can see considerably higher liver plasma ratios that are sustained over the first 60 minutes of treatment with methotrexate in the transgenic mice. Now, as we've seen with a lot of genetic loci for pharmacogenes, they get pretty complicated, and for a non-genetic specialist like myself it gets a bit hairy, but The interesting thing about this particular gene locus is it's located on the short arm of chromosome 12, but there's a whole run of other related transporters genes located next to it, and for reasons that don't often seem a bit surprising, some of these, the orientation of these genes, these related transporters, are different. So SLCO1B1, you can see the arrow indicating the direction of transcription, same as in 1B7 and 1B3, which is right next to it on the gene locus. But SLCO1A2 is in the opposite direction, the gene's opposite configuration. This is not that unusual in genetics from what I can gather, but just seems a bit strange. Okay, so you can see it's a pretty big gene. It's got 15 exons. All of these little blue bars here are all exons, and the light blue indicates the ones that are untranslated, the untranslated regions on the 5' and 3' end of the gene. And this is significantly susceptible to genetic variability, this particular locus, and including large deletions and partial deletions. And some people have a deletion of the entire 1B1 gene, they don't have it at all, and they also lose 1B7 as well as much of SLCO1A2. And then there are also people with inherited partial gene deletions which just truncate the 1B1 gene partially, but you have half of it, but it's still usually functionally compromised. Okay, so there's a lot of variability in human populations at this particular locus, and like I said, it's only in recent times and that PHAR, pharmacogenomics variation consortium, an international body of scientists and clinicians, have started to try and bring order to this crazy area of science and bring standard ways of naming allelic variants to SLCO1B1, because for a long time people in the P450 and enzyme pharmacogenomics field didn't talk to the people in the transporter field, so they never came up with a systematized standard nomenclature. But thankfully that is happening now, and we're starting to see these consistent star allele naming systems. And they've got their work cut out for them, because there's over 190 common variants of SLCO1B1 with a frequency of either over 5% in population. So that's a lot of variability occurring at significant frequencies. They have a big impact on the level of expression of this protein on hepatocytes, and they have significant impact on the influx of pharmaceuticals, but also endogenous substrates such as bilirubin. We'll come back to that in a minute. And the significance of this, the sort of takeaway message of this lecture today is that for some people that lack of ability to import drugs into the liver really does run them at risk of not being able to metabolise drugs and detoxify them. So these people are more vulnerable to drug-induced toxicity because they can't clear the drug from the systemic circulation. So there's a greater risk of peripheral organ toxicity. And the classic example we're going to focus on today is statin myopathy that occurs more frequently in people with variant forms of this gene. Now before we get to that though, and the pharmaceutical implications, let's just talk about a clinical condition associated with impaired handling of an endogenous molecule. So this is bilirubin, and it results in a condition known as Rota syndrome, and this can occur due to inherited variants in both 1B1 and 1B3. And this is quite similar to, if you remember way back in first semester when I was talking about phase 2 drug metabolism, I introduced some conditions known as Crigler-Najjar and Gilbert syndrome. And remember that these folks have a inherited deficiency in UGT, uridine diphosphate glucuronosyltransferase, the UGT 1A1. But and so those folks have higher levels of exposure to unconjugated bilirubin, which is bad news because it's toxic. So they have issues with early onset neurological issues, very severe in the severe Crigler-Najjar syndrome. Gilbert's is a lot milder. Now, the interesting thing about Rotor syndrome is it's kind of similar superficially. These people often have hyperbilirubinemia, so yellow coloration of their skin in Caucasians is a classic sign of it. But what's going on here is a little bit more complex. So think about the role of what bilirubin is. It's basically a metabolite of heme. So heme, of course, an all-important iron-containing protein, very important in haemoglobin, in all sorts of cells, including red blood cells, but it's a toxic substance. So when macrophages break down erythrocytes and chew them up, they rely on a couple of enzymes, heme oxygenase and biliverdin reductase, and one of the products of this biochemistry is bilirubin 9A alpha. And after this is formed in the macrophages, it's then excreted from the macrophage, makes its way to the hepatocyte, where it gets glucuronidated to form bilirubin diglucuronide. And this is the one that folks with Crigler-Najjar and Gilbert syndrome are deficient in. But what happens in the case of this other condition, Rotor syndrome, they can carry out these or that chemistry, but there's a normal efflux of bilirubin glucuronide by MRP3. This is a ABC transporter, efflux transporter in hepatocytes. And there's this process of recycling whereby there's a normal reuptake of bilirubin diglucuronide, which is carried out by 1B1 and 1B3. And this is the one that's deficient in folks with Rotor syndrome. So it's basically involves this mild sort of hyperbilirubinemia, nowhere near as severe as Crigler-Najjar, but still a significant condition for some people, because if you're dependent on a drug that depends on this pathway, then you've got to treat these people carefully. Now, so what is it that gives rise to Rotor syndrome? What are the genetic deficiencies involved? Well, it's got a lot to do with two dominant SNPs, single nucleotide variants, really. And this is something we highlighted in earlier lectures. Often there are haplotypes in pharmacogenomics where there are two alleles within a gene that are quite dominant in populations, and you can get different combinations of them due to genetic recombination. And so what happens in this case, we get this allelic shuffling, and there are basically four significant haplotypes due to shuffling of these two alleles, one at position 388 in the gene, where there's this two purine variants forms, adenine and guanine, and then at position 521 there's a thymidine and cytosine variants. Okay, so there's a number of four different possibilities here due to recombination during sexual reproduction. So there's an A388 T521 variant, which is basically the wild type, 1B1 star 1. There's a GT, recognised as star 37 allele, a 388A 521C variant known as star 5 allele, and then a G388 C521 allele known as star 15. Okay, so these are prevalent in human populations, and their frequency varies according to ethnicity and geographical distribution. So why are these particular alleles important? Well, we can see what goes on in them is that that variant form at 521 in the gene, which involves that T to thymidine to cytosine substitution. This is pretty significant because it changes the amino acid from a valine to an alanine, and this is a missense mutation. So basically, there's no it's a null mutation. There's no catalytic activity in that particular variant. People with these variants. The same holds true for this other site within the haplotype, position 388 in the gene adenine to guanine. This results in an asparagine to aspartate substitution. That's a pretty significant one. They're quite different amino acids, even though it's hard to remember their differences in their name. They're quite a significant different amino acid substitution. This also is a missense null mutation, so this results in significant reduction in transport, many drugs. Okay, so here we can see these common non-synonymous 1B1 SNPs that occur at this SLCO1B1 locus, and they involve these different combinations of these 521 alleles and then the other allele at this other site, at least in that one, sorry. If you want to go online, you can have a look at this nice cave. This is a comparative allele viewer, and this is a nice schematic way of keeping track of the substitutions occurring at different sites in the critical 1B1 gene. And you can see that for some of these star alleles, there's a number of variants that can occur across the locus, and those that are with functional significance have this symbol here with circle to square transition, telling us that they are functionally significant, some of these variants. Once again, as we've seen with some of the other P450 variants, there are significant differences in ethnic distribution of SLCO1B1 alleles, and we won't go through this in too much detail. This hasn't been as investigated as thoroughly as cytochrome P450 of polymorphic variants, I think, on a population level. But what we do have, even though the numbers are a bit small in terms of subjects, you can see there's some significant differences across populations in the prevalence of these different alleles at SLCO1B1. So the white is the wild type, and you can see different proportions of the population, very prevalent in some of these Northern European populations in the Middle East, but then much less common in Sub-Saharan Africa and even East Asia, where the STAR37 allele is much more frequent. Okay, so we can't assume that everyone has got the same metabolic capability or the same transport capability with regard to 1B1. There's variation across populations. Now, people have looked at the functional impact of having these different variations, and the interesting thing is that it affects, you can pick this up with a number of different approaches, including a Western blotting approach with an antibody against these different forms of the protein. And you can see that there are differences in the signal. You get this truncated protein if you've got this, if you are heterozygous for this particular variant form at position 521 in the gene. You can also see it with restriction fragment length polymorphism. So this is during PCR and looking at digesting the gene with a restriction enzyme. You can see clear differences between people who are homologous homozygotes for GG relative to heterozygous or homozygous AA. Okay, so what are these variations doing? Well, that V to A substitution occurs in a membrane spanning domain, transmembrane domain here, position 174 in the protein. And then the other one that's important is, we're highlighting in this lecture anyway, occurs out here in this extracellular loop, position amino acid 130. Now, why does all this matter clinically? Well, we'll just look at that topic now for the rest of the lecture. Why does it matter whether you have one of these wild type or allelic variant forms of SLCO1B1? Well, it's got a lot to do with, it has a big impact on your response to statins. Now, statins are a little bit of a controversial class of drugs. They're a cholesterol lowering family. Been a bit of debate in Australia over the last decade about who should get them and when they should be diagnosed, when they should be prescribed and for how long, all those sorts of questions. But they can achieve significant reductions in low density lipoprotein cholesterol. So that's one of the reasons why the primary reason they use clinically for people at risk of strokes and heart attacks. But one of the reasons for their controversy surrounding their use is their propensity to cause myopathy. Now, thankfully, the severe myopathy is rare, really quite rare, but there are milder forms of it that are not necessarily rare. In fact, a lot of people will have just sort of generalised muscle pain when they're on statins, and you need to take that seriously because it can be an early sign of something more serious. It gets complicated because the statins belong to a family that's fairly been around a while now, and there's quite a few family members, and they differ according to their basic physicochemical properties. So some of them are quite lipophilic, others are quite hydrophilic. So these are the greasy ones over here, atorvastatin, fluvastatin, and then some of the more water-soluble ones, pravastatin and rosuvastatin. Most of them have a potential for causing myopathy, but it varies a bit according to the chemical individual drug. Okay, so the symptoms include muscle pain, fatigue, weakness and cramping. Thankfully, that severe myopathy syndrome, rhabdomyolysis, is rare but potentially life-threatening. So ideally, we'd like to be able to identify people who are at risk of these severe forms of myopathy during treatment with statins. And this is a famous study from way back in 2008. This is one of the great studies in pharmacogenomics history, really, and it was this effort that really put pharmacogenomics on the map in a lot of clinical settings, because it showed that you really could identify people who are at risk of this nasty myopathy syndrome. So this is a search study, so it's a study of the effectiveness of additional reductions in cholesterol homocysteine trial, and they basically used genome-wide association studies. And this is called a Manhattan plot because you're looking at the frequency of different variants, gene variants on all the different chromosomes in humans, and plotting them, and they look like a sort of a skyline from Manhattan. But you're looking for statistically significant associations with particular gene variants, and this particular study identified this SLO1B1 variant, this site 521 in the gene, that position we highlighted earlier, as being strongly associated with myopathy. And it's a significant risk factor. You can see here that people with the TT genotype have a very low risk of myopathy. So these are all people receiving 80 milligrams of simvastatin. If you've got one copy of the variant allele, then you've got a little bit of an increase in risk of myopathy, but if you've got two copies, you're highly vulnerable to myopathy. Okay, so this was 15 years ago, quite a big deal. People looked at the impact on the pharmacokinetics of simvastatin, and simvastatin has interesting kinetics. It's basically a prodrug, a ester, and it gets spontaneously hydrolyzed in blood to form this sort of ring-opened hydroxy acid form here. This is the one that's predominant in vitro. And so when you look at the levels of this metabolite in human patient, these subjects, people with that CC genotype have higher levels compared to people with the TT or the heterozygote TZ genotype. So significantly higher systemic exposure to simvastatin, the hydroxy acid, in people with two variant forms of the gene. And this affects different statins to differing degrees. So simvastatin is the most vulnerable. So the increase in statin exposure, so this is the area under the curve basically of your plasma concentration time profile, goes up about 221% in people with this CC variant, CC genotype. But the impact on other statins is significantly less pronounced. So atorvastatin is also fairly significant, but some of these others are only modest impact in the case of fluvastatin. So if we come back to that diagram, it tends to affect the greasier members a bit more than some of the others, but it's not necessarily the case. Okay, so you're probably wondering, well, what's the big deal? Why are people with this inherited variant form of the transporter, SL1B1 transporter, more vulnerable to myopathy? Well, it basically seems to mean that if you're treating someone with simvastatin and they lack this SLCO1B1 transporter, that shouldn't be a receptor there, that's a wrong name, it's a transporter. Basically, you're less likely to accumulate the drug in the liver, and you'll undergo less metabolism, and then leaving higher concentrations in the bloodstream. So there's higher exposure of skeletal muscles. So to the drug and its metabolite, esterase metabolite. Okay, so the esterase forms in the plasma. It doesn't need to drug doesn't need to get in. Or the esterase is present in the plasma to form the hydroxy acid metabolite. It doesn't need to get into the liver to form that. But then the clearance of that metabolite depends on SLCO1B1. Now you might think that people with this variant form of the transporter might also lack as good a clinical response to the drug, because the target of action for a statin is cholesterol biosynthesis in the liver. You're wanting to shut it down in the liver, but from what I can tell, it doesn't have a major impact on the clinical response to simvastatin and other statins, your SLC1B1 genotype. I don't know why that is. It's a little bit confusing. Okay, like I said, that was 15 years ago, and on the basis of that knowledge, pharmacogenomic testing was sort of implemented in a number of countries, and they began looking for people with these SLCO1B1 variants, see if they could minimise the chance of myopathy in those patients. But also what people found was that just genotyping at that site, that 521 site in the gene, you can reduce the risk of myopathy, but nonetheless there's still a lot of inter-individual variability between people in terms of their simvastatin pharmacokinetics. So this group in Finland, a lot of pharmacogenomics going on in Finland, probably because it's a relatively small population and genetically homogeneous, so you've got a lot of pharmacology going on in pharmacogenomics as well. And they also asked, well, what are the other genes predisposing people to variable pharmacokinetics. So what they did here, and this is an interesting study, Mikkonen and Associates last year, they published this study where they used, again, genome-wide association studies. But this time, they're not looking at risk of myopathy, they're looking at variable pharmacokinetics. So they're looking at screening according to area under the curve of the plasma concentration time profile. So that's a fascinating approach. I just wanted to know, what is it that, what are the genetic factors that give you a large area under the curve during simvastatin treatment? And they found, using this approach, a couple of variant alleles, some that caused a increase in plasma area under the curve of simvastatin, some that caused a decrease. And the interesting ones here is that they found a P450-304 variant. So this is in a cellular gene, position 664, again a T to C very single nucleotide variant. This causes a comparable increase in area under the curve to the SLCO1B1 512 T to C variant. You can see here, where are we, the p-value here and the effect on the area under the curve. There's this SLCO1B1 521 T to C substitution causing about a 60% increase, but they also got a 60% increase in people with this CYP3A4 variant. Okay, so that's interesting. It just shows that some people might be really vulnerable to myopathy with these drugs, because if you've got a combination of variant forms of 1B1 but also CYP3A4, you might be out here with the really large area under the curve of simvastatin. Okay, so this is showing how pharmacogenomics really is valuable clinically, because it lets us identify these subsets of patients that have the increased risk of side effects with these sorts of drugs, because not only can't they transport the drug, they also seem to have this altered capacity for metabolism. Okay, well that's probably enough for SLCO1B1. Hopefully that's giving you a bit of an appreciation for how not only a P450 drug metabolizing enzyme is important in contributing to variable responses to drugs, but also the ability to transport drugs. So in this case, this very important protein that facilitates the uptake of drugs out of the portal circulation after drugs get absorbed from the gastrointestinal tract, go into the portal circulation, and they get taken up by the liver to get metabolized. So if you can't do that because you've got an inherited 1B1 gene that's deficient in a null gene, null activity, then that might leave you vulnerable to side effects from some of these drugs like statins. So still a lot of work to be done on working out the full significance, clinical significance of transporter variation, but hopefully in coming years we'll make progress in that area as well. Okay, so that's the last of the pharmacokinetic pharmacogenes we'll look at. To wrap up this little block on pharmacogenomics, tomorrow we'll look at another area which is challenging to me to teach, which is T-cell mediated toxicity. Some drugs cause serious skin toxicity in small numbers of people, horrible conditions you can get, and it's got to do with inherited variant forms of HLA locus genes. So we'll look at some of that tomorrow. It's a bit more toxicological. Thanks very much for your attention, and I'll see you later on perhaps in the seminars. Bye-bye. See you later. Bye-bye.